This podcast sometimes uses strong language, and it may cover topics that require viewer discretion. Also, it's a podcast. It's not a primary source of information. If you'd like to follow along visually with the show, or read any of the papers we talk about yourself, you can find all of those links and more at ridiculousresearchreview.com.
Okay. ED, do you want to give us that good scientist find?
Good scientist find. We got a good one today. This one comes from The Independent, and Pat actually sent me this one. The universe could undergo a catastrophic change that could alter absolutely everything, quantum machine shows.
I like that it says everything and nothing simultaneously.
It says everything and nothing simultaneously, and it has the best 2-paragraph exchange that I have found in one of these articles so far, which I'm going to read because they're very, very short. About 50 years ago, quantum physicists suggested that the universe could be trapped in a so-called false vacuum, where it appears stable but could be about to move into a true vacuum and be even more stable. That could lead to a total change in the structure of the universe, but scientists know very little about it, including whether it would happen, when, and how long it might take.
I think this was one of those sentences where I am left with less information than I started.
This is
Correct.
Psychic damage. When RPGs have psychic damage, this is what it feels like.
I think this is
The entire article is like that.
When it comes to the simulations of quantum machines and physics and stuff like that, I don't really think anybody should read those papers. I think that's a paper for about 30 people that you do not know.
This is the first time I've ever heard James say, hey, people shouldn't read that science.
James hates science. That's the clickbait article of today.
No, it's less that, but I'm sure there's context, and I bet at the conference they're having, like, really important conversations about the state of the field and whatnot. I think the average person, and including the average science journalist, just shouldn't read this paper. There's not a point. You're not going to get it. You're not going to have the necessary context. It's like most graduate physics people with graduate degrees in physics probably can't read this paper because it's like simulation on a simulated quantum computer about physics is too many layers deep for common knowledge. It's not going to work. Do you understand what I'm saying? Like, I'm not gatekeeping. I'm just like, this is too abstract.
But this is the real scientists find here is scientists find a research paper even James doesn't like.
Oh, we get to start different this time. Welcome to the Ridiculous Research Review. I'm Pat Brennan. I am a professor of theater and game designer, and today I am at the helm. So why don't you go ahead and introduce yourselves?
I'm James Lynch. I don't really know what to do with myself because I'm going to learn something new. But I also know that Pat is a devious motherfucker, and whatever he's about to share with us is going to be jarring.
James, from now on, for every letter you forget to put on your name, you owe me $50.
James Lynch, Dr. James Lynch.
Thank you.
PhD.
Correct.
PE.
Thank you.
California license number 42303.
And that's how you know James has student debt. I am Edie Churchill. I am a operations professional. I have done some theater, and I exist and am here.
Fantastic. Thank you so much. Okay, so today I am bringing us the research that we are going to discuss. But as mentioned, I have been trusted with the helm of this illustrious podcast, and so I will immediately abuse that trust, veer us off the road, and we're going to go into some uncharted territory.
Oh boy.
Typically, the way that this works is that James finds a peer-reviewed, sourced research paper that he brings to us. I specifically do not have that. Okay.
Great.
I have.
So we've blown up the whole conceit of the podcast, but that's fine. I'm sure it'll be
thrown
good.
It away. I've thrown it out immediately.
Season 1, we've decided, actually, forget
Season
about that.
already gone.
So, what I have is a scientific research study done by, like, real good scientists. There is real good science here. We're going to get into that. I also have other research papers that are peer-reviewed that these scientists have done on a almost identical study. And where possible, I have also found primary source data. There is a whole bunch of primary sources from this, whether it be blogs from the scientists themselves to multiple documentaries.
Oh my god!
But you will notice that there is. I've already got the first slide up for our friends here, and the first slide of my project is the word of the day is skepticism, because I need you to bring an incredibly healthy level of skepticism to this, right?
Okay.
I'm not a scientist. I'm not specialized in the things that we are going to talk about today. The people that we are going to talk about, the project we're going to talk about, is not currently published for peer review. And while I have many, many sources for this information, they really run the gambit. Like, I talked about, a documentary. One of these documentaries, for example, is put out by the Travel Channel. It's not good, and you shouldn't watch it.
Did you watch it?
Hey, but they also did Anthony Bourdain, and he was good, and I just watched like 4 hours of him before getting onto this podcast.
James, I did watch it. It's an hour and a half long.
Wow.
I would say maybe generously 15 minutes are about the thing that we're going to talk about today, and the rest of it was other stuff. So, I have really good sources. I have really bad sources. What I want you two to do is have like your yellow flags at the ready. And as we're going through this, if you get a yellow flag, you just let me know when something starts to upset you a little bit. All right.
For clarity's sake, at the end of this, are we doing our scaling?
We are doing our scaling. Yes.
Are we doing it based on the science that these people did, or on you and your research project about the science these people did?
That's a fair. Hey, listen, that's fair. Well,
do we get to do peer review live? Wow.
Live peer review. Let's
Live
go.
peer review. Here we go. Okay. So what are we doing? So we're gonna go over to slide 1 now, and what you are looking at is our team.
Dudes on a boat.
Yep,
Some dudes on a boat.
One of these guys has a great Santa beard.
So the man on the far left is Professor Neil Gemmell. Neil Gemmell is a geneticist. He is the chair of he's the chair of reproduction and genomics at the University of Otago in New Zealand. Okay. He is on a boat right now with a team that he has put together, right? So the beginning of this research project is he puts a team together. We're not in a lab. He goes to a freshwater location, all right? And he is taking samples. James, that man with the beard that you pointed out, that's Adrian Shine. If we have time, I do hope to spend some time talking about Adrian Shine.
He's a fantastic man.
He is part of this team, which, Edie, that should be a yellow flag for you, because you know who this is.
Sure is.
But I do like him, so hopefully we do get a chance to talk about him. But mostly what he provides to this research is the boat, okay? He's sort of the source for the boat.
He sure does got a boat.
That's mostly what Adrian's doing here.
Any listener who's choosing not to follow along with the slides, this guy is a quintessential dude who has a boat. He looks a lot like he's got a beanie. He's got a big old beard. He looks like he's got a boat.
I don't want you to commit to this too much, because there's going to be a photo of him later, I hope, that you will enjoy much more. Like, this
Fantastic.
is not quintessential guy on a boat. I've got quintessential guy on a boat.
Fantastic.
I will also say, though, that in this image, he is standing on a boat with a couple other guys doing what can only be described as what appears to be science.
It is super duper a photo of dudes doing science.
Yeah.
There sure do be some big tubes.
We've got Neil Gemmell. We've got one of his research team. We've got Adrian Shine on this boat. They've got a big, it's probably plexi tube, right, with like metal bits on the end, and it's on a rope. So what they're doing is they're going out to this lake, and they're starting by taking water samples. And they take samples at the surface, both on the shore and in the center of the lake. And for that, they literally just, lean over the side of the boat, scoop up a couple of liters of water, right? But they also take samples at deeper levels, and that's what this great device is for. I actually really like this thing. I've never seen this thing before, but I think it's a really good example of a very mechanical solution to something that you would imagine scientists going and like probing deep into a lake with a robot or something. The way this works is if you look at the picture on the left, you'll see that the cap is open on the bottom. It's basically a spring trap for water. So they've got a 200-meter rope on this thing. They lower this thing down into the water. You can see in his left hand, he just has like a metal weight that is like donut shaped that the rope is run through. Yep. And they just basically chuck that down the rope. And when it gets to the bottom, it bangs this thing on the top, and that bottom latch is spring loaded, and so it snaps shut, and that lets them drag back up, like, I believe it's 2 liters of water that they then get to collect, right?
That's kind of great, yeah.
Yeah, it's very mechanical. It seems very reliable. I like this thing a lot, honestly.
It's a cool tool.
Personally, I would have had a little computer thing on it, but like, you don't need it. Don't have to.
You don't need it.
You don't need it.
Why? When you can just chuck a bit of metal at it.
There. Yeah, that works.
So, like I said, they get they take samples at the surface level at 100 meters deep and at 200 meters deep in and around the lake, but also in and around nearby lakes. Those are going to be their control samples. Okay. In total, they take about 238 samples. And they do this over roughly the course of 10 days. They are limited, from what I can tell, it seems, by the hours that they have access to the boat. They then take this water, and what are they doing with it? They are DNA testing it. But not the water, obviously, because water doesn't have DNA, right?
The stuff in the water.
The stuff in the water. So they're going to take all of these 2-liter samples. They run them through a filter. And this is where we start to get into some of the information that I don't have, right? So I can look at the other research paper that they did, which we'll talk about in a bit, where they do very, very similar science, and they have all of the information on there, and they say that they run it through a filter that has like 1 micron, thick sieve pattern in it, essentially, to extract the data. I am making some assumptions here. I am assuming that they are doing a very similar process here, right? Like in that other paper, they also talk about the way that they sterilize all their equipment to make sure that there isn't cross-contamination and they don't get like errant DNA readings, right? I assume that they do the same thing here, but they haven't published this paper yet, so I can't say for certain. But it's the same team doing the same process. And what they get back when they take all of these samples and they filter all of them and they scrape out the gunk, basically, they send it to a lab and they basically read all of the DNA sequences that they can in this stuff. And what we end up with is a chart that looks a little bit like this. Wow. This chart is cut down. Again, as James has said before, I'm purposely hiding some information because it'll be more interesting later. And this chart is much wider in reality, but what you can see essentially is description of where they took the water sample. So it either says lake edge or lake center. You can see the depth that they took these samples at. 0.5 meters is for the surface, and then 100 meters and 200 meters. And then you can see all of the reads that they got from this sample. So if you look at that blue highlighted sample, you can see that they had 19.5 reads of unclassified birds, but they had like 123 reads on cattle. Again, this starts to get into where I don't know too much about DNA, but this is like, the reason it is like 0.25 is like they got partial reads off the DNA sequences, right? So like, it's the first 25% of cow DNA. So they're like, yeah, that's probably cow.
Yeah, what I would say, I am curious. I would be curious to see what these numbers are, but I bet that it really is a matter of getting strings of DNA and then running a comparison to see does it fit against
what's really
duck's
odd
DNA.
About this chart that I'm looking at, that just sort of gut feeling. They seem to get a lot of DNA hits at the lake edge. And most of those make sense. Deer, dogs,
A lot of
things
birds.
like that.
Awful lot of birds.
A lot of birds, all mostly at the lake edge. There is a disturbing number of readings for humans in the center of the lake.
Sure, but what you have to remember is that this is just like, this could be anything, right? So like, this could be hair, this could be skin, this could be just like
Okay.
somebody, took a piss in the lake kind of a thing. And they can get
Yeah.
readings off of that basically. And what this mostly comes down to is, I will say, is that, like, from my understanding, is that, like, the higher the reading they get, the more likely there was, like, evidence of that thing in that area.
Gotcha.
Right?
Yeah.
And so what they do is they take all of this data, they combine all these samples together, and we end up with a pie chart that looks like this, right? So they sample everywhere around this lake, and this is everything they get. So, like, 18.7% of their sample readings are of salmon, right? It's a lot of salmon in the lake, right? You see toads, you see eels, like oyster catchers. There's a whole bunch of different.
Waterfowl, dogs.
Yep.
Yeah.
What
And
is a
there's-
stickleback?
I'm not sure. I should look that up. I don't actually know what a stickleback is. I would guess it's a bird. But,
I'm on it. Hold on a second.
I just saw a cool name and I had to know.
It is a type of fish.
It's a type of fish.
Sweet.
Oh, it's a little itty baby fish. It's teeny tiny.
So they get both land and water creatures, because both land and water creatures are in and around this body of water, basically, right? Like, the cows go in there and they die or they defecate or whatever, and like you get those reading samples from that, for example. But obviously the fish live in there as well. So they get all this reading from there. So now I do want to pause for a second, and I want to talk a little bit about the paper that we are specifically not talking about today, but which I am pulling a lot of information from. So the paper we're not talking about is titled "Environmental DNA, Metabarcoding Reveals Strong Discrimination Among Diverse Marine Habitats Connected by Water Movement." Really rolls off the tongue. It's a real nail
Classic.
biter.
Classic environmental biology kind of title.
Yeah. So this process we've been describing up until this point is called environmental DNA, or eDNA. And what it allows geneticists, biologists to do, essentially, is go to an area and essentially take a shotgun blast snapshot of all the things that there is evidence of living in that area, right? And so for this paper, they did this first paper, the one that is actually published, in 2016, so 2 years before they do the paper that we're talking about today. They do this freshwater study in 2018. The old paper, the one done in 2016, most notable difference is that it's done in a coastal area, so it's saltwater, basically. And what they are looking for specifically is to basically judge eDNA's use on a system where there is water movement, because one of their biggest concerns essentially is that because the water moves around so much, the DNA gets spread throughout the whole system, and then there's not a really good read on what's in what area. And what they find through this study is that it's actually not true at all. It's actually, this is a fantastic way to get a snapshot of an area, right? They find that, and I'm going to go to this next one. Here's some figures from that paper on the next slide. These Venn diagrams basically are the samples that they took from those areas. And so you can see up in figure A, in the sandy beach, they identified 56 different types of species, right? Only 3 of those were only found in the sandy beach. There was 3 of them that were found both in open water and in the sandy beach. The Venn diagram, crosses over further, and you see that 11 of them are found in all 4 of the areas that they sampled, basically. And so what they find is that they're able to really clearly tell what creatures are only in a specific area and which creatures move between them. And their findings line up with actual observations that other scientists have done, basically.
Nice.
Seems to check out. My question was this research paper peer reviewed?
This research paper was peer reviewed. That's why we're talking about this one, right?
Cool.
And moreover, it uses the same eDNA metabarcoding process. And from everything that I, an outside observer who only has, first party but internet sources to look at, it appears to me as though they are doing basically the exact same technique to take the DNA, right? So they're going out, they're taking samples at like surface level at like a few hundred meters deep. The only major difference is that they're doing it in a coastal region and not in a freshwater area. And so the freshwater area presumably would have less water currents, for example. So you would guess that like they didn't really find that the DNA moved around too much here. You would guess that it moves around less.
In a lake.
In
rather
the lake,
than a river or something like that.
Yes. The freshwater location we're going to talk about is both a lake and has some river waterways in it, but it's certainly not as active as a coastal region with tides, right?
Totally.
Basically, what the conclusion that they come to with this is that, like, eDNA is a really good tool for doing ecological studies, right? They say that, like, you know, the way that we would normally have to do a study like this is that, like, a scientist would literally have to go out to an area and look around or put up cameras or something. With eDNA, they can essentially do work that would take several weeks in several days, right?
Yeah. And you don't need scuba gear.
Right, that's the thing. They mention that in here. I have a quote from their notable findings where these technologies are particularly appropriate for inaccessible regions where visual surveys fall short in acquiring the necessary data. One of the things they're specifically talking about is aquatic settings, right? Where, like, you know, you have to get in scuba gear to go look around and maybe see, you know, the fish that you're looking for or not. It's not really clear.
Yeah, a lot of fish tend to hide too, so that's harder too.
Right, exactly. Yeah, they talk about a lot of burrowing creatures and stuff like that in this paper, and they say that, like, yeah, they're able to find the DNA examples of those creatures. I guess the last thing I would say with this is that the DNA that they are sampling, they find through their studies, their estimate is that it lasts about 3 days in the water. They do say that DNA could hang around longer if it was colder, but for the most part, like, 3 days is a pretty good estimate. So when you do one of these eDNA kind of like shotgun approach water samples, you're going to know everything that was sort of in the area within the last 3 days. All right.
Mm-hmm.
That's cool because you could then do, like, I could picture like a fun seasonal study. To, like, which migratory birds come through your lake, you could find out.
Right.
Neat. Cool science.
Yeah, cool science. And here's where I will pause for a second, and I will admit to you that you were probably having a question at this point, which is that this doesn't seem very weird, does it?
How are they applying this science?
It's not very ridiculous, anything that they're doing. I've brought you a bunch of scientists that are doing some in-depth water sampling. Well, Neil and team, well, we're going to jump back to them in our freshwater study with the unpublished research.
Oh boy.
Have come here with a hypothesis, right? Because they went into this first one with a hypothesis, right? Like does, or with a question, does, you know, the DNA move around in an active water system? So in our freshwater study, it might help you to know that we are looking at our samples for 4 specific things. They're trying to see if they can find evidence of sturgeon, of catfish, of eels, and of plesiosaurs.
God damn it. Yes. God damn it.
Yes.
And Neil points out that plesiosaur, we don't really know we don't have that DNA sequence.
No,
right? We don't know what that looks like.
We're not quite sure what a plesiosaur DNA looks like.
But he also states that as a geneticist, like, we know where it would fall on the tree of life. It would fall somewhere between a bird and a reptile. And if they got hits for something that
was an unknown DNA,
unknown bird, unknown reptile-like creature, that might be something that they are really interested in for. And I think that you may have started to figure it out. But what is weird about our science is not what we are doing it is where we are doing it. So let's go ahead and zoom out that chart from before.
Oh, yeah. Oh, good.
So where are we?
No, you're not. They didn't put... Oh, my God.
This is a zoomed-in map of Loch Ness.
This is a zoomed-in map of Loch Ness.
It is? Oh, my God, it is.
It is.
Okay. Look, I wasn't sure if there was... I'm sorry, I didn't see the Loch Ness. I thought it could have been Lake Champlain. Like,
it could have been Lake Champlain.
Yeah.
So...
Can I say... Like, I'm happy with my scientific impulse here because I have 2 things to say. Number one, as you're describing how this works, I understand. Like, I pulled up Wikipedia. I was curious about the barcoding versus meta barcoding. Barcoding is like trying to match a string of DNA onto a particular species. That's cool. The meta barcoding that they're doing this in parallel to an entire group of animals. So I can definitely see why they think, like, oh, well, we can look at a whole group. The weakness that I had identified in my head is that if you don't already have the DNA of a particular thing that you're looking for, this is dramatically less powerful.
Yes.
Like, and dramatically less convincing. Because... like, barcoding is really cool if you have a full genome. Like,
Right.
if you don't, bunch of stuff shares genome. We share like a massive portion of our genome with tomatoes.
Right.
Like, this is a hard thing to do. And they're like, plesiosaur, sure, it's somewhere between birds and reptiles. Why not? Let's go. Yeah, sure.
So yeah,
Yep.
so let me talk you down off a cliff a little bit in a couple of different ways. So you are looking at this map. This is the full web page of all of the data samples that they've pulled.
Oh, so they put up the web page?
They did, yeah. All of as they went along, everything was publicly available. And we're going to talk a little bit about the website in a little bit, but unfortunately, the website has lapsed because they couldn't afford to keep paying for it.
Sure.
And so Neil, in his talks, basically says to use the Wayback Machine to go and find the website that they had built for all this stuff. So the primary source says that this is the way that you can go and access it, basically. Also worth noting that this is... The data that is available is not representative of all 248 samples or 238 samples, whatever it was. It's not representative of all of it, which, let me tell you, is a point of contention on some less than scrupulous parts of the internet. Let's leave it at that.
Okay, yeah, that's awesome.
Yeah, they're pretty unhappy that not all that data is there.
Yeah. They're like, oh, the man's like, it's out there.
They're hiding
the truth
there.
is out there.
Right.
Nessie's hidden in the secret data.
But again, let me talk you off this cliff a little bit, because I know that you're seeing a map that has all these cute little plesiosaurs on it. That's not them saying this is all the evidence of the Loch Ness Monster.
Yes.
That's them saying this is where we took a sample. Okay? It's just a
And they
cute
just
little...
chose to use a cute little plesiosaur because they like Nessie. They're into this because they like Nessie. They're having a good time.
In fact, we can pause for a minute here, and I can jump ahead a little bit, and I can say that Neil Gemmell would agree with you, James, on a lot of this.
I'm sure he's a good scientist and he knows the limitations of his tools.
Yes, and what he came out here with was a hypothesis, because if you remember, what I said was that they were looking specifically for catfish, eel, sturgeon, or plesiosaur, because what Neil Gemmell is not trying to do is he is not trying to prove the existence of the Loch Ness Monster.
No, he's trying to prove the existence of something far more interesting.
Loch Ness Monster 2, Electric Boogaloo. No, he's asking the question, like, look, throughout quote unquote history, and I say that in quotes because like the Loch Ness Monster craze really kind of gets started in like the 1930s. There have been lots of, supposed sightings. So the question is basically, what are people seeing? And it wouldn't be an episode of the Ridiculous Research Review if I didn't provide you with some photos that are a little bit upsetting. So we're going to go over to the next slide.
Oh, no.
And so just to give you an example of how big these creatures can get in fresh water, on the top left, that is a sturgeon. Top right, we've got some eels, freshwater eels. Then we have a catfish. And then just for comparison, because I didn't realize how small they actually were, a drawing of a plesiosaur.
I thought plesiosaurs were way bigger than that.
Wait,
so did I. I thought
that's unbelievable. They were...
Yeah,
they're not that big.
They're not that big.
They're not that big.
Yeah.
They're smaller than a giraffe.
Yeah, they are smaller than they're much smaller than a giraffe, I think.
I thought at least giraffe size. I thought it was
I was
like
thinking
giraffe
like
size in
whale
the
sized,
water.
honestly. I thought they were big motherfuckers.
Yeah, I think we all did, mostly because of media and things like that. But no, they're not really.
What's the Loch Ness horror movie? Hold on.
There's a couple, I think.
Yeah. Those are definitely big. Those are...
Yeah, the movie monsters are huge. But look at that catfish. That catfish is as big as me.
I'm more afraid of a big catfish than I would be of a plesiosaur.
Yeah, honestly, that catfish is horrifying.
If that catfish swam up next to me, I would get the fuck out of the water, and I would be like, I saw a monster.
Big boy.
That catfish could swallow half of you without trying, You're just like, your feet are getting digested. It's terrible.
Yeah, so, when we get down to it, Neil is not trying to prove the existence of the Loch Ness Monster.
Ah.
Actually, what Neil is doing is Neil is trying to sell the public on eDNA. That's really what he's doing out here.
Oh, okay. Yeah.
And he has a couple of really fantastic talks where he talks about this. One of them in particular, a talk he does with Genomics Aotearoa. It's about an hour long. I don't recommend that anybody go and find any of the documentaries about this, because there's a documentary that the BBC put out, there's a documentary that I mentioned that the Travel Channel put out, and honestly, there's like 15 minutes of science, maybe, and then there's like an hour plus on like all the like admittedly cool but completely nonsense history of the Loch Ness Monster, right?
Yeah.
So like, if you want to learn about like the surgeon's photograph, or any of that stuff, like you can learn all about how that stuff is like definitely fake, but it's not good science, because when it comes down to it, the science portion of this is takes about 15 minutes to sort of get through, which is that Neil and his team go out, they do a lot of like comprehensive, well, maybe not comprehensive, but they do a bunch of DNA study, basically, on the lake and take a snapshot of what's living there. And what they come back with is, we did not find any evidence of a big monster. We did not find any evidence of catfish. We did not find any evidence of sturgeon. But what they do find is they find a lot of evidence of eels.
Lots of eels.
Interesting.
If we jump back to that pie chart, you can see that about 6%, about 6% of the DNA that they sampled was eel DNA. So these things are all over the place.
Okay.
And Neil kind of puts forth at the end that he's like, well, it could be like a really big eel that people saw. And let me tell you, there's a lot of folks on the internet that are very upset about the suggestion that eels could get that big.
But again, also, the plesiosaurs weren't big.
Well, so there are some people who get upset about the idea that eels can get that big. There are also some people that get real jazzed about it.
And I don't like either of them. Neither of them are invited to my dinner parties. But like, if we look at this eel photograph, right, like it seems as though sometimes eels travel in a group. Again, I'm not a scientist. I can't say that for certain. But like, here's a photograph that I have from the internet, which is real from all sources that I can, reasonably look into, and it shows, I don't know, like 20 eels all swimming together. And if
doesn't look like AI to me.
And if I don't know, even if like 6 of these big guys were sort of like swimming along the surface, that might look like a really big Loch Ness monster to you. So like Neil kind of hypothesizes that it's like some of these sightings could be eels. But that's kind of all he can say. What he can say definitively at the end of this research, and what he's very careful to say, is that like, yes, there are eels in Loch Ness, excuse me.
Are we coming back to our good buddy yet?
Yeah, we can talk about our good buddy for a second. So, one of the folks that he does this with is a member of the Loch Ness Project, Adrian Shine. Here's a great photo of Adrian Shine.
Oh, can I describe this guy?
Please.
He's sitting on his boat in a gray-green, just classic tailor suit. Looks like the most Scottish boatsman I've ever seen. Like, oof. He's the rich guy who lives by the water with a very nice boat and a very nice suit and a big old beard, and that's great. It's a great photo. I wish I had a photo of myself like that. That'd be amazing.
So what I can find on Neil is that Neil honestly seems not insidious or harmful, right? Like, no. He is a local who seems to just really like Loch Ness and the area. He's part of the Loch Ness Project, which is sort of just like, I don't know, like a conservation society basically associated with Loch Ness, essentially. He describes himself as a naturalist. He does not actively dissuade people that are looking for the Loch Ness Monster. And in fact, their, visitors center, for lack of a better word, essentially has history about the Loch Ness Monster and things like that. But obviously that's because that's what tourists want to come and see. But like mostly he sort of, from what I can tell, sort of like pushes people in other directions, where somebody would be like, oh, Nessie this and Nessie that. And he'll be like, yeah, and we also have great waterfowl in the area, basically. Or like, check out the fish that live here.
There's good birding here, actually.
Yeah. He does appear, I will say, in many, 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 many of these documentaries. He's written a handful of books about monsters and sea serpents that seem to be from a, let's call it hobbyist anthropological point of view.
Sure.
I'm not saying that he's like a good source or good science. I just don't think he is as insidious as a lot of people are. And I'm going to pause for a second here because I think that we do need to talk about the elephant in the room for just a second, which is cryptozoology, right?
Love it. Love it from
From an entertainment standpoint.
It's great.
Yeah.
Right.
So,
If you're ever in Portland, Maine, the International Cryptozoological Museum is there. It's great. Honestly.
It's pretty cool.
Really fun. It's mostly a collection of somebody who's got a whole bunch of faked footprints. But that's entertaining.
it's fun pseudoscience, and it's pseudoscience because they don't really follow the scientific method, right? I mentioned before that like the Loch Ness monster stuff kind of gets going in the 1930s, and like that's when like the craze of it all starts. That's actually kind of true of all cryptozoology. It mostly gets going in the early 1900s and kind of hangs around, and we get like, you know, Bigfoots and UFO and all of that kind of crops up in the same area. That's when it kind of gets popular. And it is rightfully derided because it doesn't use the scientific method to reach its conclusions. Mostly it deals in belief, hearsay, and sort of expeditions that like are going in with foregone conclusions, right? So because of that, I would say Neil's study is not a cryptozoological study, right? He is not looking for the Loch Ness monster. He's looking to see if he can find 4 examples of things living in the lake. If one of them happens to be a plesiosaur, that would be wild, right? The Operation Deep Scan, the other thing that you're seeing on this slide, that's sort of where Adrian Shine kind of gets his claim to fame, because like I said, he does not really dissuade folks from the Loch Ness monster stuff. He plays into it because he's a local. He loves this, loch and the weird monster stories that are here.
Right.
In 1987, there's a little date on the photograph, or on the drawing that you're seeing here. He basically gets a bunch of boats equipped with sonar, lines them up in a row like that, and does like a sweep of the entire loch trying to get a ping on the monster. And Adrian and many of the folks that were there that day would say that they did not find anything, and cryptozoologists would say we got 3 suspicious pings.
Yep.
But the thing about sonar, especially sonar in 1987, is that like it's not the cleanest image, and moreover, sonar does tend to interfere with itself. So if, for example, you lined up 20 boats with sonar and drove them up and down the loch, they might cause a problem. In fact,
You might get a few errant pings.
in fact, with Operation Deep Scan, they actually had to turn the settings of their sonar on all the boats to the lowest setting, because when they first fired it all up, they couldn't see shit because all the boats were interfering with each other.
Yeah, that's great.
So, and like,
Oh my god, we were the monster all along.
I love this drawing. I love this photo. I love that like they put this together. Like, I think it's great fun. But when it comes down to it, it's great fun. And I think that is sort of where cryptozoology needs to live.
Mm-hmm. I will also say, in everything I've seen of Adrian Shine, that does seem to be his MO, that it is just great fun for him.
Yes.
Yeah.
Is he somebody who like pops up on? Is he the type that like pops up on the History Channel documentary? Who's got like a wink and a grin a little bit, whereas
I would imagine
he's
so.
not
Yes.
He's not the credulous guy who wants to say that all paleontologists are liars.
Right.
No.
Right.
And in my research into this, I did bump into like several folks that have like, masters or doctorates in, for example, sociology, and then are using that to claim that they are a doctor and that they are looking for monsters.
Nice.
Adrian isn't that. He doesn't, claim to have any sort of like degree behind this stuff. From everything that I have seen, and he's done a lot of stuff. I'm sure maybe he's, been he misrepresented himself or something. I don't know. But like from everything that I have been able to see, he very much presents himself as just like the guy that lives by Loch Ness, and he likes it here.
As a descriptor of what this man looks like, though, if he claimed to be Charles Darwin reincarnated, I would believe him.
It wouldn't be too far off. The last thing I guess I want to say is that I would be remiss if I didn't mention that the three of us, and me particularly, do love cryptozoology, perhaps because of our upbringing. We all grew up in the Bridgewater Triangle, the quote unquote cryptozoological capital of the world, which I didn't learn until recently was a phrase that wasn't coined until the late 1970s.
That's right.
Interesting.
Super, super, super recent.
Same guy that cryptozoological museum.
Yep.
Was founded and is the collection of the guy who coined the Bridgewater Triangle. So, like, it's all connected, Pat.
And I know that to be true because he shows up in the travel documentary about this research study.
Ah, nice.
So, the world of cryptozoology isn't, super large. There's not a lot of people that they can get to talk about this stuff, I think, for right reason. And so that kind of brings us to the end of this research experiment and the why of the question of, like, they did good science here, as far as I can tell, right? There's lots of sources. Like I said, Neil Gemmell gives a talk that is an hour long and absolutely worth your time. It's super interesting. And a lot of what he goes into is why this project happened and why he did it, basically. Because he admits right off the bat that, like, this very much could have been a career killer, right? Like, he is the chair of a fairly respectable department at a fairly respectable university in New Zealand.
Right.
He is a geneticist with a number of papers under his belt. And putting something like this out into the world, I could see it, like, severely tarnishing his career. But like I mentioned before, he doesn't view this as a search for the Loch Ness Monster. He really views this as science communication. And in fact, I can tell you for certain that his university is on board with this, with members of the University of Otago calling it the best piece of, like, public science communication that has ever been done, basically.
Wow.
And essentially their PR department, who sort of, like, tracks all of this stuff, love that he has done this because it gets everybody, including us right now, talking about their university and the science that they're doing. He tells a fun little anecdote of how, the conversation of whether or not to do this started on Twitter, basically. And he, put out into the world, he saw something about the Loch Ness Monster, and he was sort of like, hey, like, did anybody ever use eDNA to, like, see if they could just find something down there, basically? And it gets picked up and spread around through, like, tabloids and then real news sources, basically, and it moves really quickly. He becomes basically viral overnight.
Wild.
And
that's a wild way to have that happen, where the conspiracy theorists pick up your idea and then it turns into funding. But it captures the public imagination. And he talks about the fact that the reason he decides to do it is he goes to pick his son up from school and finds himself swarmed by a group of 8-year-olds that all want to talk to him about this, and realizes that if he goes and he does this research project, he might show even just one of these kids that, like, science is a career in a field that you could go into, and that he could inspire them to become a scientist one day. And like he says that he's like, yeah, all of this other stuff is great. He decides he talks a lot about the monster effect and how really he uses the Loch Ness Monster as bait to get funding to go and do this. Yeah, hell yeah.
Yeah, but the inspiration for doing this is basically like, whatever. He's like, I could go and I can sample the lake and I might find some stuff about ecology, but in a bigger picture, I could inspire one kid to go be a scientist one day. And that would be worth the entire trip, basically. Yeah.
I mean, hell fucking yeah, man. Getting funding to do fundamental science research is so hard. Like, even when you don't have, like, the headwinds that we're facing, like, today with, like, NSF programs being just shuttered because they mentioned the fact that women might do science. Like,
right.
It's even before all of this bullshit, it is so hard. And like, extra if you're not in the U.S. It's so hard to get funding. If you need to, like, put Loch Ness as your logo for on your publicly available website,
if that's what gets you attention for your data,
if that's what you need to do, I'm all fucking for it. That's great, because what this means that you get to use these tools. People hear about it, both the public, and I'm also even thinking about like I love that note about like inspiring somebody else to do science. I think too about like the people who are like looking for tools to more ways to study the environments they're interested in. This is a very cool tool, and maybe they hear about it for the first time because somebody did it in Loch Ness. You know,
Right?
You've talked about this before, James, but like so much of these research papers is marketing.
a lot of it
to,
is, yeah.
to gain attention for your work, for your university's work, to bring in that money. I do have to say though, Pat, there's a piece of this that you're missing that I'm not sure if you're done or if you're going to get to, and so I have to ask, how far down the eel hole did you go?
I went a ways down the eel hole. What I'll say is I went so far as to find another paper.
Can I just register a complaint for the phrase "the eel hole"? So I don't want to hear about
acknowledged
eel
by the
holes.
board.
Just like register.
Does the board recognize the complaint?
Can we just like
Heard.
put that on the record? I'm not in favor of the term. Continue.
Yes, I went so far as to find another research paper that I'll admit that I like read through, but like I did not, go and check the sources on that and things like that, where essentially somebody was checking the math on how big eels could statistically get, which kind of seemed suspect to me, basically. But they basically were
Right.
saying that like, finding an eel that big was like an astronomical chance.
Well, see,
and so
that's the thing. People have built on this particular research study saying that there are eels in Loch Ness to come up with a hypothesis called eunuch eels.
Oh yeah, eunuch eels. Sorry, I do know about eunuch eels. So
Yeah.
Here, allow me. So specifically,
I don't know what this is. I'm here for the ride.
I'm sorry. I should have talked about this. Yes. Yeah. So Neil and team, and several other scientists out there, basically, like the question gets asked, like, could it be just like one really big eel, basically? And there's a hypothesis that if the eels don't breed, like if an eel just like doesn't breed for whatever reason, it might just stay in the loch. Ignore that next part of its life cycle and just continue to grow and become enormous.
Interesting.
And so
okay.
a let's say very debatable and not well studied or evidenced hypothesis for the eel theory is that there are eels in Loch Ness that are just not getting laid and are just getting really big instead.
Yeah. And so the theory being that perhaps the Loch Ness monster was actually just a really big eel.
It always comes back to incels, doesn't it?
It always does. It's always incel gym bros who are just getting really big and not getting laid.
Yeah. This is the hypothesis that Adrian Shine presented in the piece from the BBC that I saw.
Interesting.
I will say he ended by saying, like, this isn't a thing I believe to be true, but it is kind of fun to think about.
Okay.
Yeah, that's there's from what I was able to tell, and I admit I did not extensively search the eunuch eel theory, there is not much scientific support for that idea.
That's great. That's great. We are going to link all the videos and this slide deck so we can share this excellent photo of Adrian.
Yeah, so I will link the slideshows, and I will link as much of the primary sources as I can. I will even go so far as to tell you where you could see an hour and a half long documentary that you probably shouldn't watch. But I will link to that talk as well, which you absolutely should go and watch. But so this is a great transition point, because this has sort of been my difficulty in sort of putting this episode together, was finding primary sources and sources that I trust. It was great when I could find an article that Neil Gemmell wrote. It was great when I could find a talk that he gave, but so much of this has been caught up in all of these other nonsense sources. And that would be fine if the paper was out. But the paper isn't out. And at the end of his talk, Neil Gemmell mentions that they do still intend to publish this paper, but that talk was given 2 years ago. So it takes a long time to do these things. I understand that. This is certainly not the most pressing of whatever else he's doing, because I think from his perspective, this project has really already done everything that he wanted it to do. The data of a snapshot of Loch Ness is interesting, but I think science communication was the point, which I think we need to take into account when we are judging the ridiculous versus the research level of this. And I wish that I could point to a summary in the paper where they say the point of this research study was really science communication. But all I have to say for that right now is talks that he has given where he has said that exact thing.
I think, honestly, that might be part of why it's hard to publish. Because, because while a single sample on a particular week or of Loch Ness is interesting, but one of the things that journals tend to want is interesting, but then also impactful and generalizable in some sense.
You're hitting on one of the big flaws with cryptozoology as well, which is that it is generally difficult to prove the non-existence of something. So what a lot of the naysayers on this Loch Ness project basically say is like, well, you only went and sampled on these 10 days and Nessie just wasn't there that day.
Yeah, totally. Yeah, totally.
There's a ridiculous point in one of the travel documentaries where somebody leans over to Neil while he's riding around on this boat, and they're like, well, have you heard the theory that, the Loch Ness Monster, like, I don't know, shifts between dimensions or whatever? And to his credit, Neil is really good at controlling the conversation. You can tell that this is a man that has spent some time in education, because his response is basically like, well, now we're starting to get into like the area of belief, and that's not where I work. Where I work is science. Right. So I can tell you whether or not we find example of a catfish, basically, but I can't tell you whether or not I believe Nessie shifts through dimensions. You'll probably need an astrophysicist for that, basically.
Right.
Nice.
It's so well handled, and absolutely not worth watching an 1.5-hour documentary for, but it is very well done.
Yeah.
So we do have to get to our ratings here.
We do.
And I would say, based on both the idea of sort of science communication, and what I think seems to be some pretty strong application for climate studies and sort of populations of fishies, I would start this around a 3 for impact.
For impact?
Hold on. Sorry, for impact?
Yeah.
For research.
Yeah. I like this as a 3 for research. Like the other publication is also great. It's a very useful tool in the useful tool in the tool chest. And
I think eDNA's got some room to grow.
Yeah. I mean, when was this paper published, Pat? Did you say? When was the other one?
Well, so again, the paper in our talk is not published.
Right.
But the other one.
The other paper... Hold, please. The other paper, the study was done in 2016. I believe the paper came out in 2018, but I'll have to double check that.
Yeah, I feel like there is something impactful in the work that is being done. I feel like Neil seems to have come from a good place for it, with his idea of like it being using the Loch Ness as a way of gaining attention for this science study that he was doing anyways.
Yeah.
I feel like a 3 is fair.
And hey, now we know there's a shitload of eels in Loch Ness. That's and I didn't know that before.
You do now.
I do now. More information. Great.
All right. So then, how are we doing on ridiculous?
Weirdly, I don't think this one's that ridiculous. It's kind of ridiculous. But even the way that it's phrased, where it's like, here's 4 things this could be, including plesiosaur. Like, it's just including plesiosaur that is the thing that's a little ridiculous.
I'm delighted. I knew you all thought I was going to zig, and here I am zagging, which is, I tried to find a paper that presented as a little ridiculous, but I think it's actually very, very reasonable. I think it's
I think
basically just good science.
I think it's thoroughly reasonable. I again would put it like right in the middle on the ridiculous scale, like a 3 again of like.
Oh, I don't think it's that high. Do
I'd
you?
go a 2. I'd go easy.
I'd go a 2. Okay. I'm that easily swayed on it that it's a 2. Yeah, I like. Because it feels.
To be fair, right? Like, he is using the premise as bait. He says that a lot. Right? Like, he knows it's a little bit ridiculous, right?
But even the premise is like, it's just run of the mill crazy. Like, oh, you believe in the Loch Ness monster or Bigfoot? Like, okay, yeah, cool. I've met that crazy like 6 times on Tuesday. Like,
yeah, right.
So just to clarify, final score, we are saying 2 for ridiculous, 3 for research.
Correct.
Yeah.
I guess the last thing I'll say on this is that I will admit that what I did with this paper felt a little cryptozoological in nature, which is that I did not naturally come across a paper that was about the Loch Ness monster. I remembered cryptozoology, and then I was like, surely someone has done science on this. And so sure. I did, when I was putting this episode together, I did have my answer first and then go and find the paper that supported me.
Oh, so you did. You did cryptozoology, but for the paper.
Man, I'm gonna have to like, I will cut this, and I don't have time to talk about it. But like, I learned so much about Loch Ness for this project. Like, it is a really interesting place. Honestly, like, it's super deep. It
Loch Ness is super cool.
A crazy amount of water. It's like so full of peat that you can't see. So it's like a great place for a monster mystery to exist because it's just kind of spooky. It's like cliffs on all sides, and then like super deep dark water in the middle. Like, it's great.